And I'm going to run through the aspects of the lab protocol that we have here, and I'm also going to sort of talk through the materials and how to do that sort of thing. Okay? All right. So for those of you guys who don't know me, well, you guys all know me because you all watch the videos, but I'm Mr. Matthew. I'm one of the honors bio teachers here and have been forever. But I'm going to walk you guys today through the rate of photosynthesis procedure for the term 2 lab. So just a couple things before we get into it. The goal of the term 2 lab is for you guys to get an experience of trying to execute a fairly easy, fairly simple lab experiment and generate your own data that you can do some intro analysis of. That is the goal. It is not supposed to be to find 10 crazy variables or all kinds of crazy things. So you're going to do something very simple. You are going to get some closed containers. So these are the kinds of closed containers, and I have a giant box of these down in 254. And you're going to basically get, well, that one's not very good because it doesn't close, but you're going to get lids like this, and you're going to get 12 of these closed containers. And 6 of them you are going to make your control, and 6 of them you are going to make your test. All right? And what we're going to use as our model organism in the system is we're using Alodia. This is the same plant that we used for our photosynthesis model CER that we did at the beginning of the year. And so what you'll end up seeing is that we'll have plants here. Now, a couple of things about these plants that we'll notice is that they have these like offshoots and they have these sides and stuff like that, and they're also not all the same. So some of them are just like little tiny tips of them, and some of them are like super, like super long and that sort of thing. So you're going to need to come up with a way of standardizing the plants that you're dealing with. And I'm going to recommend that you do one of 2 things as you're standardizing of your plants that are in there. One is you can do it by weight. So you can dry these off as best you can, get a piece of paper towel, rest them on there, get most of the excess water out of them, put them on a balance and take an initial mass and know what the mass is in each of the cups, and therefore you can figure out the photosynthesis or the amount of dissolved oxygen per mass. That is one way to do it. The other is to do it by length of plant, and literally by that what you can do is you can say, well, we've got a stem that's this long, and then for these ones that have an offshoot you can say, well, the main stem is that long and then the offshoot is this long, and you can measure it using a ruler, and therefore you can compare the lengths of the stem. You don't have to do both. I would just pick one. And this is something that you can use to standardize cup to cup, because the likelihood, and I know that sometimes there's protocols that say cut down the stems, that also could damage them. Some would be damaged, some wouldn't. Are you cutting them in equal ways? I would say either pick by mass, which will be tough because they're pretty light and there's water clinging to them, or by length, which is also tough because they're awkwardly lengthed and they're not all quite the same. But one of those 2 should be a standardizing way. So you should know in cup 1, my plant has these characteristics, either this length or this mass. In cup 2, my plant has this length or this mass. And that way, when you get your oxygen rates for each of them, you can divide by that standardizer. Does that make sense? Okay. So we're going to do 6. We're going to do 6 cups that have that are going to be tested on. The other 6 cups are going to be your control. So your control can be one of 2 types of control. You can either do a positive control, in which you take 6 plants and you put them in there, and again do the same exact thing, but you're not going to apply the test condition to it. Or you could fill them up with the equivalent amount of water, okay, and then expose that to the same test conditions as the plant. Again, this is your choice. But you want to have something either as a positive control, that would be the plant in 6 cups, just in room temperature, not, everything exposed except for not doing your variable, or 6 cups exposed to the same condition. Okay, so one with water, with 1 plants and 1 without plants, or 1 exposed to the condition and 1 not. Difference between a positive and negative control. Okay. Does anyone have a question about positive or negative control? All right, you guys are chatty. All right, so we've talked about the allodia. We talked about the closed containers. What is the experiment you're going to do? Well, you're going to see how does photosynthesis change with temperature. And so the way that we're going to do that is we are going to use these heat mats. These heat mats, we have several of them, okay? And we are going to decide to change the temperature, and we can control the temperature of these. Now, the temperature, we basically adjust the different heating mats in here, and then there's a controller that goes with that, and those controllers will allow you to change it from 70 degrees Fahrenheit up to 85 degrees Fahrenheit, which is also between 20 degrees Celsius and 30 degrees Celsius. I will tell you that 20 degrees Celsius, 70 degrees Fahrenheit is roughly the temperature of the room. So I would recommend that you pick a temperature that is elevated by either 5 degrees Celsius, meaning try 25, or maybe go up to 30 degrees Celsius, but you will only change to one different temperature. And again, we'll have some different mats, so you will be able to take your containers and we'll have, I think, 4 different mats, and we're going to set those up either under a light in 258 or we're going to set them up in room 254, and we'll have them out on the counter, and they'll all be in one location. Okay, so again, what's a variable you want all your tests under? You want them all under the same temperature, right? That would be there. If you're doing a negative control, you'd want your negative controls exposed to that same temperature as well. All right, and then the next thing we have is our dissolved oxygen probe. So you will be using the lab quests and the dissolved oxygen probe just like in the CER lab, and you will be recording your milligrams per liter of dissolved oxygen in your containers. You're going to want to do this a few times. So you're going to want to get obviously day 0, and then you're going to want to get them over some amount of time. And it's going to be up to you how many days do you want to run that, and you want to do it on a fairly regular basis. I'd say because we're starting so early, some groups could get set up as early as next week if you wanted to set some up. The problem with that is we do have the Thanksgiving break, and that's a problem. You very easily could do this over a 5-day period, like a Monday through a Friday, and then just go and quickly take the measurements, alternate who does it every day or every other day. You can either do it every day or, again, over 5 days or over 10 days. I would recommend you block out at least 5 days to take the temperature over minimum. I probably wouldn't go over 10 days as your first start, but if you want to do day 0, day 5, and day 10 and see if your numbers come out nicely, that would be something you could start before Thanksgiving break, and that would work. Or you wait after break and then just take them every day or every other day over the week after break, and that would work as well. Okay? And then lastly, aged tap water. I will have a bottle just like the one that we had out, which will have aged tap water. As we've mentioned before, tap water is chlorinated. You could run this whole thing with tap water, fresh tap water that doesn't have that. You just know that you're adding chlorine. Again, as long as you're doing the same thing to every single cup, that's great. So I will definitely have a couple of containers of aged tap water sitting over on the counter that will have naturally dechlorinated by light. So they're equivalent. And again, I'll have 2 of these. If for whatever reason you drain this because you set up next week or whatever, just fill the thing back up with tap water, put it back on the counter where you find it over 254, and that way it'll be ready the next day for whoever comes along. And that's what we always try to age it at least 24 hours. I probably will set up an extra jug of it in another room stashed away for the inconsiderate group that forgets to refill it, and then the next group sets up the next day and is all mad. But we'll have a couple of extra containers, so you'll do it. And you'll notice these are not very big. They only take, I think these are 80 ml containers. So something like this, which is, close to 4 liters, I think it's like 3 liters. There you can, like 4 or 5 groups could easily set up all their containers with this right now. Yes?
So we have one positive control, which is room temperature.
It's one possibility. Your positive control could be plants set at room temperature.
Mm-hmm. And then we have one other, different temperature, or do you have multiple?
One temperature. That's a good question. So you have one test, and that's a raised temperature of some amount.
Right.
But you could also do a negative control, which are cups with just water in them exposed to the same thing, because do you know what the dissolved oxygen rate of aged tap water is at that high temperature? The answer is you probably don't know. So we do need to see, what happens to dissolved oxygen? And I will say that temperature does affect dissolved oxygen rate of just water. So that will change. So the question is, how much of that change are we seeing because of the plant versus how much of that change are we seeing just because of the chemical properties of water being exposed to heat? In term 3, we're going to let you open the box and ask a much more broader question and increase your variables. At this point, what we'd rather have you do is pick a single variable that is in a reasonable range. And by the way, these temperatures are reasonable range. And then generate some data that you can do some